Hello everyone, myself Dr. Pollobi Duara from Department of Zoology, Majuli College. I am going to discuss about the structure of nucleus. During lockdown period in India due to pandemic COVID-19, the Zoological Society of Assam has taken this initiative to help the students of zoology providing with online lectures. My presentation is a part of it. In this slide, it is shown that after going through the entire PowerPoint presentation on structure of nucleus, students will come to know the following main points. They will understand the structure of nucleus, the components of nucleus, and the parts of nuclear membrane. Students will come to know the structure of nuclear pore and transport through the nuclear pore complex along with the models of translocation mechanism and assembly and disassembly of nuclear pore complex. And they will come to know what is nuclear lamina and what is nucleoplasm. Nucleus is a membrane-bound structure that contains the hereditary information, a really very important element of the human body, mostly covered by every biology subject departments all around the globe. It controls the growth of cell and reproduction. It is found in most eukaryotic cells except red blood cells, and it acts like the brain of the cell. The shape is spherical, elongated, lobular or flattened, and the number is usually 1, it may be 2 and more. The location is in the center of the cell. In animal cell, it is in the center of the cell. But in a plant cell, the nucleus is located more on the periphery due to the large water-filled vacuole in the center of the cell. It may be eccentric, deviating from the center, or in the base or under the plasmalemma. In mammalian cells, the average diameter is approximately 6 micrometer, which occupies around 10% of cell volume. Here the structure of the nucleus is shown. The genetic material is enclosed by a double bilayer membrane, the nuclear envelope, the inner nuclear membrane, and an outer nuclear membrane. The nuclear membrane or envelope is not continuous but interrupted by some pores called nuclear pore as shown in the figure. Nuclear lamina consisting of two components, lamins and nuclear lamin associated membrane proteins, lie on inner nuclear membrane. Ribosomes remain attached to the outer nuclear membrane, and the endoplasmic reticulum with ribosomes also remain attached to the outer nuclear membrane. So, the contents of the nucleus are held in the nucleoplasm, similar to the cytoplasm in the rest of the cell. The components of nucleus are nuclear membrane, nucleoplasm, chromatin, and nucleolus. The nucleus is composed of various structures, namely the nuclear envelope, nucleoplasm or nuclear sac, nuclear matrix, chromatin, and nucleolus. The nuclear membrane forms an envelope-like structure around the nuclear contents, which is commonly known as nuclear envelope. The nuclear envelope separates the nucleoplasm from the cytoplasm. The details of the components will be discussed in the next slides. Electron microscopic studies of the nuclear envelope has shown that it is composed of two unit membranes, an outer membrane and an inner membrane. Each membrane is about 75 to 90 angstrom thick and little protein in nature. The nuclear membrane, also known as nuclear envelope, is made up of two lipid bilayer membranes, which in eukaryotic cells surrounds the nucleus, which encases the genetic material. Outer nuclear membrane often remain rough due to the presence of ribosomes on its surface. Sometimes it remains continuous with the membrane of the endoplasmic reticulum, Golgi complex, mitochondria, etc. The inner nuclear membrane contains no ribosomes, and sometimes it also remains associated with the chromatin. Further, the nuclear membrane is followed by a supporting membrane of uniform thickness known as fibrous lamina, which is 300 angstrom in thickness. At certain places, the nuclear envelope is interrupted by presence of small structures called nuclear pores. The number of pores for a particular nucleus is variable, often depend on the species and type of cells. Now, the parts of nuclear membrane. The nuclear membrane encloses the cell nucleus and consists of outer membrane, inner membrane and nuclear pores. Each membrane is composed of phospholipids arranged in a bilayer. This means that the entire nuclear membrane has 4 rows of phospholipids. Attached to the inner nuclear membrane on the nucleoplasm side is the nuclear lamina, which is a sheet of proteins that provides support for and strengthens the nuclear envelope. The nuclear lamina also attaches to and anchors chromatin, loosely arranged DNA and protein. The nuclear pores are composed of large complex proteins which allow a few molecules to permeate by nuclear envelope. The nuclear pores are octagonal in shape and have a diameter of 600 angstrom. These are enclosed by circular structures known as annuli. The parts of the nuclear membrane is shown here. The nuclear membrane, as shown in the figure, keeps DNA inside the nucleus to protect it from surrounding substances in the cytoplasm. Additionally, the nuclear envelope can regulate what materials enter or exit the nucleus. Inner nuclear membrane proteins are proteins that embedded in or associated with the inner membrane of the nuclear envelope. These are discussed in the next slides. Inner nuclear membrane protein transport is mediated by multiple mechanisms. 3 different translocation models have gained experimental support. Probably all function for different inner nuclear membrane proteins according to their unique characteristics. At nuclear pores are pathways for the exchange of macromolecules. The pores help in exchange of materials between nuclear fluid and cytoplasm. The annuli regulate exchange of the macromolecules through the pore. According to the clinical nature and size, the permeability of the pore complex depends on the size. The nuclear envelope disappears during cell division and reappears during nucleolar organization. This transport includes RNA and ribosomal proteins moving from nucleus to the cytoplasm, and proteins, carbohydrates, signaling molecules, and lipids moving into the nucleus. The entire nuclear pore complex has a diameter of about 120 nanometer in vertebrates. This is the structure of nuclear pore. First is the view from the top of the pore. The next is the view from the side of the pore. The nuclear pore complex is one of the largest protein complexes. The nuclear pore is guarded by two annuli, an outer and an inner annulus, each with 8 globular subunits, 50 to 20 nanometer diameter. Projecting inwards from them are 8 radially arranged spokes. In the center of the pore there is a central granule or plug. Such structure is called nuclear pore complex. Nuclear pore complex is one of the largest protein complexes in the cell. It provides access to the nucleus and regulates the transport of proteins and RNA across the nuclear envelope. They are aqueous channels generated from a complex network of evolutionary conserved proteins known as nucleoporins. By restricting access to the nucleus and separating gene transcription from protein translation, eukaryotic cells have evolved to control gene expression in a highly regulated manner. Nuclear pore complexes are considered to be the gatekeepers of the nucleus and facilitate almost all transport between the nucleoplasm and cytoplasm. A cartwheel view depicting half of an nuclear pore complex is shown. The nucleoplasmic filaments are organized in a basket-like structure that projects from the nuclear pore complex towards the nucleolar interior called nuclear basket. The cytosolic filaments face the cytoplasm. In addition, the obtained images reveal that the nuclear pore complex contains a structure resembling a basket that extends into the nucleoplasm and filaments that extend into the cytoplasm. A high-resolution structure of nuclear pore complex in which the position of each individual protein is resolved is a formidable goal for structural biologists. Despite its enormous size, the nuclear pore complex is made up of only about 30 nucleoporins that are largely conserved throughout eukaryotic evolution. Three models have been suggested to explain the translocation mechanism: affinity gradient along the central plug. This model emerged as a plausible explanation as a result of in vitro measurements in yeast and vertebrate demonstrating the presence of an increasing affinity gradient in nucleoporins moving from the cytoplasm to the nucleus. Brownian affinity gating and selective phase. The Brownian affinity gating and the selective phase model both predict that molecules that directly interact with the Fg repeat nucleoporins can diffuse through the nuclear pore complexes. Now, the functions of nuclear pore complex. Nuclear pore complexes are indispensable for cell function and are at the center of several human diseases. Nuclear pore complexes provide access to the nucleus and regulate the transport of proteins and RNA across the nuclear envelope. They are aqueous channels generated from a complex network of evolutionary conserved proteins known as nucleoporins. NPC provides an important control point for the regulation of gene expression. The nuclear lamina, which is a dense fibrillar network inside the nucleus of most cells. It is composed of intermediate filaments and membrane-associated proteins. Besides providing mechanical support, the nuclear lamina regulates important cellular events such as DNA replication and cell division. Nuclear lamina consists of two components, lamins and nuclear lamine-associated membrane proteins. The lamins are type 5 intermediate filaments which can be categorized as either A-type or B-type. The nuclear lamina lies on the inner surface of inner nuclear membrane, which is shown in the figure as I and M. Nuclear envelope proteins that are bound to the lamina include nespirin, imerin, lamina-associated proteins 1 and 2, LAP1 and LAP2. The lamin B receptor LAB and MAN1. The lamin B receptor transcription factors that bind to the lamina include the retinoblastoma transcriptional regulator RB, germ cell-less GCL, sterol response element binding protein SREBP1, FOS and MOK2. Barrier to autointegration factor BAF is a chromatin associated protein that also binds to the nuclear lamina and several of the aforementioned nuclear envelope proteins. Heterochromatin protein 1 HP1 binds both chromatin and LBR on the O and M, that is outer nuclear membrane. The composition of nuclear envelope. The nuclear envelope proteins that are bound to the lamina include nespirin, imerin, lamina-associated proteins 1 and 2, that is LAP1 and LAP2, the lamin B receptor LBR and MAN1 as shown in the figure. Transcription factors that bind to the lamina include the retinoblastoma transcriptional regulator RB, germ cell-less GCL, sterol response element binding protein SREBP1, FOS and MOK2. Barrier to autointegration factor BAF is a chromatin associated protein that also binds to the nuclear lamina. The functions are: it is involved in most nuclear activities including DNA replication, RNA transcription, nuclear and chromatin organization, cell cycle regulation, cell development and differentiation, nuclear migration and apoptosis. Specific mutations in nuclear lamina genes cause a wide range of heritable human diseases. It also participates in chromatin organization and it anchors the nuclear pore complexes embedded in the nuclear envelope. The nucleoplasm, which is a type of protoplasm and is enveloped by the nuclear envelope. The nucleoplasm includes the chromosome and nucleolus. The nucleoplasm has a complex chemical composition and it is composed mainly of the nuclear proteins, but it also contains other organic and inorganic substances such as nucleic acids, proteins, enzymes and minerals. The common nucleic acids of the nucleoplasm are the DNA and RNA. The nuclear proteins of the nucleoplasm can be categorized as histones or basic proteins and non-histones or acidic proteins. It may contain many enzymes which are necessary for the synthesis of DNA and RNA. Enzymes are DNA polymerase, RNA polymerase, NAD synthetase, nucleoside triphosphate, adenosine deaminase, nucleoside phosphorylase, 3-phosphoglyceraldehyde, dehydrogenase and pyruvate kinase. It also contains cofactors and coenzymes, ATP and acetyl coenzyme A. It contains small lipid content. It also contains phosphorus, potassium, sodium, calcium and magnesium. The main function is to support the chromatin material and nucleolus and providing rigidity to the nucleus. Here, a brief introduction of nucleolus and chromatin is given. The nucleolus is the largest structure in the nucleus of eukaryotic cells, which is best known as the site of ribosome biogenesis. And chromatin is a complex of DNA and protein found in eukaryotic cells. The main function is packaging long DNA molecules into more compact and denser structure. It will be discussed in the next presentation. So, after going through the entire PowerPoint presentation, we can summarize as follows. The nucleus is a membrane-bound structure which contains the hereditary information, and the components are nuclear membrane, nucleoplasm, chromatin and nucleolus. Here in this slide, the suggested reading of books and research articles is shown. Here the students can go through the following books and literatures for further references. At last, I would like to thank everyone. Thank you.